How exactly does CRISPR-Cas9 insert new genes? I understand the very basics of how Cas9 enzyme cuts off a gene with a guide RNA to help it locate. But what's still unclear to me is what we do with the DNA breaks. If we let the cells repair it by themselves, will they not just remake the segment we just cut off? If we insert a new gene, how exactly do we deliver it? Does it come with the Cas9 and guide RNA complex? Or do we use another enzyme to deliver it separately? I've just started learning about this topic, so I'm sorry if my question seems very basic. I've tried searching online but so far I've only gotten things like, we insert the gene. We deliver the new gene, or the new DNA segment is delivered. Without ** it was delivered. The answer, when you insert the enzyme and guide RNA, you also add a bunch of copies of short DNA sequences that match the region being cut, but that include the change you want to make. Then when the cell repairs the break, it's likely to grab one of these synthetic sequences to serve as the repair template, and boom, change made. Follow this channel for more Santa Explains.